So this is week 16, and we're doing a review of the digestive system and male and female reproductive systems in preparation for the exam 4. So this is the evening review session on the Monday evening, and we also have the review sessions coming up on the Wednesday morning at 10 o'clock and Wednesday evening at 8 o'clock. So those are the sessions that we have available for going through the study guide, which has been uploaded also into your Canvas course. So let's have a look what we've got lined up for this session. And I'm trying to make it as quick and efficient as possible, but make sure that we cover everything we need to look at. So as always, I have some objectives that we'll go through. I think that a lot of these are similar things. Nothing much has changed from the previous exam week's formats, with the exception that, of course, the material is slightly different. So I can't believe that we're on the second last week of the course. I'm actually very excited seeing all the amazing work that you've done. Sometimes we get so busy when we're working that we don't actually get to see how much good work has been done. We just see what's being done that week. But when I look back, you guys have really worked hard. I know that there are for some few assignments that are still missing. So please, 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 this week and definitely next week, it's time to get those in. I realize that things happen, so I'm trying to be flexible of those, but don't leave them at the very last minute, because I do have to get the grades in. So we want to make sure that we get everything finalized on this course. But overall, I'm very, very impressed of the standard of work. So we'll have a look of this week's material. And then we've got the next week, which is the finals week. And we've already mentioned in previous sessions that there's going to be finals week project that you'll be completing. And I'll provide good details for what to do with that at that time. So as with the previous weeks when we have had exams and lab practical assignments, remember there is no new assignments this week, but this week is really the time to catch up on any assignments you're missing. I really recommend that you go to your grade book on the Canvas and look through what is missing and are the grades what you expected them to be. Sometimes there's a little bit of challenges with the transfer from the Connect from the publisher's side to the Canvas. So I want to make sure that you have points recorded everywhere where there should be points, and they're the points that are correct. This is the time to make sure that that all is up to date and in order. Also make sure that you get those assignments if there's anything missing. It's better to submit in something than not get points at all. You guys know at this stage that I'm always generous, trying to come up with at least some points, even if the answer wasn't exactly what I expected. If you tell me something else, then tell me something else about the structure, the area, the process, and I can at least award partial points. This week we have the exam 4 and lab practical 4 as the new elements. Last time we had quite a lot of issues with the lab practical 3, but now everything should be running. I think that we should be in a good place with that. Remember, both of these are due by the end of this week. So on the Sunday evening, or just before midnight, 1 minute before midnight, they are due. Of course, as before, I will try to work with you, but please, 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 if you can, don't leave them to the last week. If something happens and you have to complete it early next week, I will work with you, but try to get these in as much as you can as early as possible. And we will be on this session focusing on the exam 4, since you have good study guides and you are very aware of what's going on the lab practical 4. You've seen a lot of those questions in your practice for the lab practicals on each week. You also have like I said, the study guide with blanks for the exam 4. So if you haven't had a chance to download it or print it for yourself yet, this would be a good time to do so, because we will start looking through it in just a few moments. Of course, if having the study guide is not the way you learn, you do what works for you. But the study guide that I provide for download or print, it has blanks. So we're going to go through those blanks tonight. And also for the lab practical 4, like I said, there's a good bunch of lists of the terms and structures that you should be able to identify. You also have a chance to do the practical practice version. I really encourage that you do that and do it multiple times so you get to see all of those questions on that pool. Of course, I realize that the pictures that I have on this video are from the previous exam weeks, but they are similar buttons, similar documents. So practice the lab 4 practical multiple times, so you have a chance to see all of the possible questions that might come up. For the lecture slides, I think if they're not up, I'm just in the process of uploading those, not the exact ones that I use in my video lectures, but the ones from your publisher's site, so matching with the textbook, so the content is very, very same. Again, if that helps you, those are there for your resources and assistance. I have been very open to do retaking assignments, or if you need something opened up, I'm also with that. Totally happy to facilitate that, but do remember, send me very, very clear details, which assignment, and best way to reach me is really through the Connect messaging. That's a system. That's probably the best place for getting a hold of me and making sure that your request reaches me as quickly as possible. I'm just going to add a note here. So make sure to send me that message if it comes too close to the time when the final grades are submitted. I will not be able to facilitate new submissions. So this week and early part of the next week is really the time to get this done at the latest. Like I said, this week you don't have new assignments, so I think that I would personally try to get it done. Now it's a good chance to get those finished. My advice would be to try to get something submitted for all of the assignments and then go back if you want to retry something else. Remember, there's also extra credit assignment available as part of the finals week project. And just finally, we've seen already about the Cranium Cafe, the tutoring service that's available through the college. And you can do it fully online. in addition to that, I'm of course available in this for this course. So all you have to do is let me know and we'll organize a one-on-one meeting. So what I'm going to do now, I'm going to jump back here to exam 4 and change screens. And we're going to look through the study guide that we have for this week's material. Or I mean, previous chapters, but this week's exam material. So I should have a study guide ready for you here. And last time I was able to get back to making notes and other stuff. I think that we're at the right place now. So we should be able to make a little bit better job with highlighting also what's important. So we started the material for this set of chapters on the digestive system. And we remember that digestive system was again linked to everything else we've seen so far. So digestive system is really what our body does. So what we do is we convert sugars into ATP. And that process requires a little more, it requires for this conversion to happen, we need also oxygen, and as a waste product, we are left then with the carbon dioxide and water. So all this happens on a cellular level at the level of mitochondria, which I'm sure you remember from your basic review of the cell organelles. And I'm going to do very rough drawing here. It looked something like this, and there were those cristae inside membrane folds that played an important role. So I want to make sure that you know that it's not just your respiration on the whole body level, it's also at the level of the cellular level of the mitochondria that plays role. But the big thing to notice here with this chapter kind of intro is that as a waste product, we had the carbon dioxide and water. They were transported as carbonic acid, and then as a bicarb, we were able to eliminate a lot of that through respiration. So that's something that we've already had a look. Extra hydrogen ions, we urinated through or eliminated through urine. So all these systems that we've had a look, they do link together. So we keep that in mind, refer back to those, find those overlapping parts. And here comes the really important stuff that I really, really, really want you to be familiar. You know that I feel rather strongly about this, that these are concepts that everyone should know. There are 4 essential functions for the digestive system, of course, taking the food in, breaking the food into manageable sized chunks so that they are small enough chunks that they can actually cross that membrane to enter to your body, not just travel within the digestive tract, but actually physically enter to the bloodstream or to the lymph if it's large fat molecules, or to the interstitial space, wherever these enter from the digestive system. And of course, we also eliminate the material that's not digestible. And that's going to become our feces. But the big thing here is these 2 terms. So we have digestion. And what digestion was, as I'm sure you remember, digestion was the process of this food, these large food molecules being broken down. And the reason why we have to break them down is that we need to make them size that we can actually absorb, that it's small enough to cross those membranes, whether it's a membrane at whatever part of the digestive tract, but without doing first this digestive work, breaking down a large molecule into smaller molecules, we cannot move on then to have those small molecules crossing the membrane and becoming available for the body. So I really hope that you make sure that you know the difference between digestion and absorption. That would be an essential concept, and one of these, if nothing else, learn these kind of things. So digestion, breaking down and taking in the large food molecules, making them smaller, and absorption, when we can then have these small molecules to actually cross that membrane from your digestive tract into your body. Because if you think our digestive tract is just a tube inside us, opening from one end and opening from other end, it's still technically the part that's connected to the outside world. It's not inside us as such. So that's why we need to do both. There were two types of movements that were really important for us to be familiar with, that I had emphasized to you, and these kind of keep coming up. We've seen them in few other chapters already, and these are terminology that you want to be familiar with. So we had the concept of peristalsis, which was this wave-like movement. As you can see here on this diagram, the food moves along the digestive tract in a manner that when one part of the tube contracts, it pushes food onwards, and then another part right next to it contracts, that pushes the food further forward, and this synchronized contraction works ahead and helps us to transport this food along the way ahead in the digestive tract. So this really moves the material along the tract. So that's an important thing to remember about peristalsis. There is movement, and it's clear to what direction we're moving. If we jump to look at the other kind of movement type that we see, we had also segmentation within the digestive tract. And segmentation really didn't generate movement as such. What segmentation did instead, it helps us to break down whatever is in that tube at that very spot. And it also helps to mix whatever is in that tube at that very spot and in the nearby area of that spot. So rather than this synchronized progressing movement generated in peristalsis, segmentation is like squeezing in one place and one place only without actually materials moving significantly. So there's a little bit of back and forth movement, but really the big deal there is breaking materials, squeezing those materials to break. And that helps us also to mix food that's within the digestive tract. So those are the two things that I would really like you to be comfortable in terms of the types of movement regarding this chapter. We also saw the digestive tract had certain layers that we always tend to find within the tract, and they keep repeating no matter at what part of the digestive tract you are. We tend to see these 4 layers, or tunicas, if you wish to use that terminology. They keep repeating in all parts of the digestive tract. What's interesting to me is the digestive tract is essentially just a tube, and it's a tube, like we've said, with similar layers at all parts. At some parts, this tube grows bigger, sometimes it grows smaller, but still at the end of the day, it is one tube. And then we had, of course, a couple of accessory organs, which we will cover at the very end, so liver, gallbladder, and pancreas. But let's look at this general structure of the digestive tract wall. And we're going to start with the very innermost layer. So you remember the center of the tube was called lumen. So this layer is right against the lumen, and this layer is known as mucosa. So what's the function of the mucosa? This layer protects the other layers underneath it. This layer helps with secretions passing through the actual lumen of the tube, and this layer also facilitates the absorption of materials through it. And if you look, you will notice that for that very reason, there's a lot of folds, a lot of villi, a lot of microvilli to provide us as much surface area for this absorption. In terms of secretions, what's interesting is that the glands are not actually located within the mucosal layer. The glands are located rather within the next layer, which is called sub-mucosa. So within sub-mucosa is where the glands are located. The ducts of the glands opened through the mucosal layer. So remember that the submucosal layer houses actually those glands. In addition, within the submucosal layer, we have there a lot of blood vessels, a lot of nervous innervation, so a lot going on within that layer. The following layer, working our way from the center of the tube outside, is gonna be the muscular layer. And we'll notice that the muscular layer actually is rather thick on this diagram. And we're going to notice that there's two types of muscle fibers. The innermost ones are circular. So those facilitate the movement to that direction. And I'm just trying to change the color of my highlighter. And the layer after that, that's outermost, is gonna be longitudinal layer of muscles. So we have both of these layers present in the digestive tract at all parts. There was one oddity, one additional thing to note, that only within the stomach, but within stomach we can find a third layer, and this third layer was oblique layer. So in oblique layer, the muscle fibers go to third direction. So stomach's function was to mix up material, and that's exactly what this facilitates us to do. It helps us to make movements to various directions within the stomach. And the very last layer, that's the outermost layer, when we work our way from the inside of the tube to the outside, is gonna be called serosa. And as you'll notice on this diagram, serosal layer is not super thick, but it is still an important layer. So I'm just gonna emphasize it here, but I want you to know that although it's not a thick layer, it's just as important for us to know what it is and what it does. So serosa is made of a lot of connective tissue, and its function is to anchor this digestive tube to its site so that it doesn't move around. So those were our 4 layers of the digestive tract that we see in all parts of this tube when we work our way through it. Another kind of a introductory concept that I do want to mention, and I want to make sure that we all know, is the term peritoneum. And we've heard the term peritoneum throughout this course on multiple occasions. We discussed that some structures are retroperitoneal, other structures were subperitoneal, or intraperitoneal. So we use peritoneum in the abdominal pelvic cavity to locate structures, and we describe the structure in relation to this layer. So I just want you to be sure that you know what we've been actually talking. And the peritoneum is this layer that lines the abdominal pelvic cavity, goes around the our small and large intestines when you take a cross section, and also anchors the stomach on its place. So that's where the peritoneum is and where it's located. And there's one part of peritoneum in particular that's worth of knowing if you're doing dissection work or ever end up having a chance to observe this fold here, which becomes visible when you go through from the anterior side through the abdominal area, through the belly. You first cut through the skin, then adipose tissue, then muscles, your rectus abdominis muscle, for example. What you end up noticing is that there is this big flap-like structure that loops right here in the front, and that's called greater omentum. So that's one anatomical landmark that I would encourage you to know. Just very, very brief introduction about the neurocontrol of the digestive system. You remember that for that, we are going to be focusing on this parasympathetic branch of the autonomic nervous system. So this was the branch rest and digest. So at this stage, when our parasympathetic branch of the autonomic nervous system is active, we are not really doing anything. We're just chilling on our place, and we're letting these processes that are not vital for running away, fighting for our life, to be more active. So a good comparison would be after a Thanksgiving meal, you probably sat down, and that's when your parasympathetic branch of autonomic nervous system was active. So you were simply digesting a lot of the food. A lot of the blood gets directed to the gut area. Our urinary system works is more active, filtering the blood, removing what shouldn't be there. So after meal, relaxing. The other branch was the sympathetic branch, which was when we have to fight for our existence, either by having a fight against the potential threat or running away from it. And at that stage, digestive system would be depressed because we don't have to worry about digestion or urination at that very moment. We're worrying about staying alive. So other systems activate at that when that branch is active. Let's have a look at the couple of basic things. In terms of the blood supply to the digestive system and from the gut area in general, all of the blood at large, with the very few exceptions, from the abdominal area, from the around the digestive organs, travels first to liver. And the function of liver is a large list, but one of the functions is that it helps us to store energy from that energy-rich, sugar-rich, nutrient-rich blood that we have been able to produce at the abdominal area. The nutrients, energy has passed to the blood, so that blood all travels through the liver, and the purpose of that is to, and I'm trying to find my pens again. Sorry about that. That's not what I wanted to do. This looks more like it. So what we end up noticing is that if we have here our liver, and this is a very quick sketch, so apologies for it not being artistically the most perfect one, but this is our liver here. And here's all of our digestive system. For the sake of clarity, I'm going to change the color. Let's do, for example, this. let's imagine that there's our stomach, and from our esophagus running up a little bit, and from there, from our pylorus, our small intestines, the first curvature, duodenum, then jejunum and ileum, and those connect to the large intestines, cecum with appendix, they traveling and transverse and descending and sigmoid colon. So all the blood from this area of the intestines travels first to the liver. So that's why liver cirrhosis, for example, can cause issues or other liver issues with individual not having enough energy because nothing gets caught up from the blood, or less gets caught up from the blood that's from the abdominal area, from the food. The connection between here bringing the energy and other resources to the liver, and connection of blood vessels and leaving the liver. Over time, if your liver starts to fail, they grow closer together and we almost end up bypassing the liver. So that would not be ideal. So for this reason, the liver plays a major role. And that's the reason why blood flows in. What we end up noticing, that the cells in the body really are kind of greedy and silly. They just, if they have a lot of energy available, they burn that energy off. They go crazy. They don't think of the future. So that's the reason why blood flows through the liver. We're going to talk a lot on this review, and that's going to be high emphasis on the test as well, about the oral cavity. Sometimes also known as buccal cavity, but probably most familiarly called mouth cavity. Or mouth. So remember, there's many different terms, but we're all still talking about all of this same area. And one of the things that we discussed very early on our review of the digestive system was that where does the digestion really starts? And often we might end up thinking, okay, when the food enters to the mouth, there's salivary amylase and other enzymes within saliva, and they start breaking down things such as starch. And then you remember things travel further down the esophagus to the stomach, small intestines, large intestines. But I would dare to claim that just the sight, smell, or thought triggers for many of us the production of the saliva. And that production is for us then already digestion, because we're producing those things that are going to break down the food material that enters to our mouth. So I would dare to argue that digestion starts with just the sight, smell, or thought. So that's a concept to consider. Functions of the mouth, we of course are focusing on the food aspect, but also remember it plays an important role in communication through speech. Also, the food doesn't just go down to our digestive system. We do a lot of screening of that food. So does it taste okay? Does it smell okay? Does it feel texture-wise okay? Because we want to make sure that we're not really eating that's something that we shouldn't, something that can be dangerous, poisonous, or so on. That's why evolution has developed a lot of sensory processes within. And we had a look of the boundaries of the mouth, most anteriorly, your lips, laterally, your cheeks. If you didn't have your cheeks, food would fall out. Same anteriorly, if you didn't have lips, food would fall out. Superiorly, so at the top, your palate. And remember, you had a hard palate more anteriorly and soft palate more posteriorly, and soft palate then changed into uvula at the very end, the hanging projection at the back of the mouth cavity, oral cavity. So hard palate is hard because there is bone within this tissue, separating the nasal cavity from the oral. And later on, it's just going to be soft tissue, so it's more posterior, part of the top of the mouth, roof of the mouth. Bottom of the mouth, the floor of the mouth, inferior part is largely taken by the tongue. If you observe it, you'll notice the tongue is much bigger than we often give it credit. Certain stores sell cow tongues, for example. It's a lot of muscle, so it's considered as fairly good to be used for food in certain parts of the world. At the very back, posteriorly, for the boundaries of the mouth, we of course have the pharynx, and part of the pharynx that's located there is gonna be the oropharynx, because this is now next to the oral cavity. Higher up, we would have nasal cavity, and further down, and that would be called nasopharynx, and further down, then laryngopharynx. But we're looking at the oropharynx now. A couple of words that I wanted to say about the tongue. Remember, forms the floor of the mouth. It's made of skeletal muscle. There's a couple of, it's not a single one muscle. There's a couple of groups of muscles, or multiple groups of muscles. Some of them are intrinsic, so play a role in the shape of the tongue. Some are extrinsic, playing role of the position of the tongue. And really, what happens in the mouth? We break down the food by chewing it, grinding it, biting it, tearing it, and we also mix it with saliva. So when we have this food that has been physically, mechanically broken down and mixed with saliva, you'll notice that we form kind of a nice, more of a ball-like structure that's easier to manipulate. If you try to swallow something very dry, let's say like cornflakes, without any milk, without any liquid with it, without chewing them, mixing them with saliva, it's really hard to swallow. It's really coarse for your back of the oral cavity, going down your esophagus, further down. So we want to do this mixture, mixing it with saliva, and what we produce is called as bolus. So bolus is this when food has been mechanically broken and mixed with saliva. We no longer talk about food within the digestive tract. From now on, moving further, we talk about bolus. Of course, there's many other functions that tongue could play a role, initiating the swallowing, speech, taste, sensory organs, and so on. Couple of structures of the tongue that I wanted to note with you. If you remember, let's imagine that this is our tongue looking from the up down, some from superiorly, and at the bottom on here we have anterior part of a person. Here we have posterior part, so the person would be looking at this way. This would be the nose and eyes here. So what we'll notice on the tongue, there's various different kinds of small rounded projections. Some of them are a little bit bigger here at the back, but we've got a bunch of different kinds of projections on the surface of the tongue. These were our papilla. So that's a term to know. We also notice, if we look now of the mouth, and I'm going to scroll somewhere else, we'll come back to that soon. If we look here at the mouth, you'll notice that there's a connective tissue band here underneath the tongue. There's another connective tissue band here connecting the gums and lips, and there's one here at the very top as well, connecting the gums and lips. These are known as our frenulum. So there's frenulum within the tongue, within the between the gums and lips, and they are a structure to be aware of. We might have to cut the frenulum if there's a struggle with the infant latching. So that might be something that we could do early on to help the infant. We already mentioned the uvula at the back of the mouth hanging there. Let's have a look what else we should know. We should talk about salivary glands for sure. So where does the saliva come? We've got major extrinsic salivary glands, and there's 3 of those. And then we've got a bunch of minor salivary glands within the oral cavity. We're not going to spend time on those, but I do want you to know the extrinsic salivary glands. You remember that saliva already played a role in breaking down substances, so in digestion. So the salivary amylase was one of the enzymes we end up finding in the saliva. So that was all about breaking down starch. We produce a lot of saliva, about 1.5 liters in a day. Remember though that all goes back to the body. We reuse it, so it's not like we're losing that. But what I want to do with you guys, I want to cover the 3 extrinsic major salivary glands that we really should know. And you'll notice here a picture of an individual. Here is the anterior. This person is looking to the front, and this is the posterior side of them, the back side, eye, nose, and mouth. So let's start with our parotid salivary gland. And often I make little rules to help me to remember things. And how I remember the parotid salivary gland is that it's located where a parrot would be sitting if a pirate would have a parrot on their shoulder. I think that in most movies and so on, we see them carrying a parrot on their shoulder. So it's located here, right next to the ear, above the masseter, facial nerve, cranial nerve 5 runs through it. So we might have to worry about that if we do operation to the parotid salivary gland. And the duct from the parotid gland, notice how it opens next to the second upper molar here. So it also is one of these glands that's located elsewhere and opens up the secretions in another place through a duct. Secretions from the parotid gland are serous, so very liquidy, very fluid. So I think that that's a good start for our review. And next, I want to talk with you a little bit about submandibular salivary glands. So the name here is a clue. So mandible, I highlighted with wrong color. That's not what I intended to do. This looks better. So it's located actually not underneath the mandible as such, but within the mandible. So I would say, I'm just trying to correct what I messed up here in terms of the colors. It's good. And this looks better. And it's located more of, I meant to say, kind of in the middle of the mandible, medially to the mandible. And we have a duct from there, which opens then at the base of the tongue, at the base of the lingual frenulum. So you remember the structure, the connective tissue band that we just had a look of. Secretions from this salivary gland are actually mixture of serous and mucus secretions. So that's something to notice, that we're kind of moving now to a different kind of secretion. And I'm just going to circle this. I didn't mean to circle it with blue, so I'm just going to highlight it with red. The last salivary gland that we should have a look of is going to be our sublingual salivary gland. So as the name would suggest, it's located below the tongue. So I have drawn it here as such. So what sublingual salivary duct does, its secretions are very mucous, so very thick in this time. So that differs a little from what we've seen. And there's many ducts coming out of it, so it opens up in multiple locations within the oral cavity. So that's something to note there. And just for the sake of clarity, if I get the pen to work, I'm just going to go back and add this duct that we saw for our submandibular here, because I drew on top of it. So those would be our 3 salivary glands, 3 structures to know. And we also had the minor salivary glands, but we're not going to cover them. Many people on this course were thinking of going to fields like dentistry, fields that involve the head and neck, oral cavity. So I'm spending a little bit more time on this. And this is something that I've taught in the past also more extensively. So I mentioned that learning the teeth and which teeth and what type of teeth we have, you can make your life easier. And the way to do it easier is that you divide the mouth into 4 parts, so into quadrants. And the order and types of teeth that we find in each are then repeated on each quadrant in the same way. So we're going to start from medially, the most anterior teeth, and work our way then to the laterally and posterior. So we notice that the very first teeth that we have are these flatter chisel-like teeth that are ideal for biting things. Like think of biting an apple. So they serve a very good purpose for that. And you'll see that there's 2 of those in 1 quadrant. Following that, we've got these sharp teeth. Some people have them more pronounced. You'll notice that a lot of the time with Hollywood stars, they get those sanded down a little bit. These are known as our canines, only 1 of them per quadrant, ideal for tearing things. So that tells us a little bit about our evolutionary past as well. Moving further back, a bunch of 2 teeth known as premolars, also sometimes as bicuspids. So they're before the set of 3 full molars, molar teeth. Molars are ideal for crushing and grinding stuff. If you look, they have a large surface that's easy to hold stuff in, very powerful at that area of the mouth, thanks to the muscles that we have there. So premolars are a little bit preceding that, and we transition to that. So those are the teeth to know. We also had a look of the cross-section of the tooth. Remember that crown was this part that we can see above the gum. Underneath the gum is going to be our root. Within the crown, we see enamel layer, which was one of the hardest materials in the body. Majority of the teeth is then going to be dentin. And then we have, of course, our central cavity where the blood vessels, nerves, connective tissue, other stuff is there. If you have ever had a root canal done, we have to get to the central cavity to do that. So that's why we've got that long needle going in and we have to drill to get there. So we want to get to that nerve within the teeth. Within the root of the teeth, we had to note, we have cementum layer. I think of it as cementing the teeth to the socket within the mandible. And then we have periodontal ligament. After that, does the same thing, anchors the teeth to its site. We continue our journey and we get to the pharynx. Pharynx is made of skeletal muscle layers. Externally, inside, innermost layer is stratified squamous epithelium. We're not going to be too interested in the nasopharynx. We've already discussed about that in another chapter. But for the digestive system, of course, at the back of the mouth, we had our oropharynx, and further down, laryngopharynx. So these two parts of the pharynx are shared with the respiratory system. Nasopharynx is fully dedicated to the respiratory system. The fancy word for swallowing, deglutition, was a term that we were wanting to use. And we've talked about the importance of correct terminology. Everything you write into medical records has potential or does become a legal document. So you want to have your terminology right. You want to appear professional in case you have to later on refer back to this. So deglutition is the term for the swallowing. I often ask my students, is swallowing voluntary or involuntary? Well, it starts as a voluntary act, but once the swallowing has been made, you cannot stop that from happening. It continues as involuntary. Oesophagus, this was the muscular tube. There really isn't much happening in terms of the digestion within the oesophagus. Its main function is just to transport material down. We see peristalsis there taking place. So that's how it's able to move material even against the gravity. If you stand on your head, you are still able to transport material along your oesophagus. If there's no food, it's squashed closed. I think that the collapse might have been a little bit extreme term. It's not open when there's no food traveling through. It pierces the diaphragm. A couple of other structures that pierce diaphragm, of course, are aorta, descending aorta turning to abdominal aorta, our inferior vena cava. So there's not a lot of holes within the diaphragm, but this would be one of those. Tells us a little bit about its importance. In terms of the stomach, very little absorption happens within the stomach. It's more of a temporary storage pouch. We start the chemical breakdown, so a lot of enzymes, secretions are put in there to the food material. Once the bolus, the term that we have been using so far, reaches the stomach, we start talking about chyme. Once it's mixed with the stomach acid secretions, now we're going to talk about, I was trying to get a highlighter up. We're starting to talk about chyme, which is a mixture of this food and other secretions within that mixed with the stomach acids. So bolus with stomach acids is going to be our chyme. It's much more liquid, much more runny. If you've ever had the runs, diarrhea, that's more of a description of how liquidy this mixture is. We remember stomach could expand and shrink a lot. Rarely we see it fully expanded. So that's all thanks to the structure of the stomach. Ridge-like folds inside the stomach are gonna called rugae. So when you do dissection, which is something that I feel sad that we don't get to do now on a remote format, you would actually see when you cut in to the stomach, take a cross section, that there's all these folds. They're bigger than the villi that we see, and these are the rugae, allow the stomach then to stretch and shrink as needed. So we've got a couple of parts of the stomach that I do want you to know. This is our oesophagus, bringing food down. Right after that, we've got our cardia, and the part above the cardia and oesophagus, so the most superior part of the stomach, is going to be our fundus. Majority of the stomach is body of the stomach, and at the very distal end are pylorus. So we could do further divisions, for example, about pylorus, but we're not going to do for the sake of keeping things clear at this level. And then we, of course, move on to the small intestines. You'll notice that there's a bunch of sphincters that we need to know, connecting or separating really the esophagus from cardia. We've got lower esophageal sphincter. That's normally squashed closed and only opens when food travels down from there to the stomach. This is more of a physiological sphincter, not so much anatomical one. If we have that sphincter open, we can have acid reflux, so materials from the stomach might actually travel up to the esophagus. You get heartburn. That would not be ideal. That irritates the esophagus, so if that's a constant problem, you might get ulcers there, untreated ulcers, and still irritating. That can even develop into cancerous tissue. So you want to get that addressed if you have a lot of problems with heartburn there. At the distal end of the stomach, we've got our pyloric sphincter separating the stomach from the small intestines. This is a visible muscular anatomical structure sphincter that we see. A couple of other things we notice, that there's two curvatures on the kind of more lateral side, greater curvature, on the more medial side, lesser curvature. So that's probably a good start for knowing the structures of the stomach. We talked about the acid reflux already, when the lower esophageal sphincter failed and stomach acid then irritating the esophagus. Hernia is another clinical situation where the stomach goes through the hole within the diaphragm that's only meant for the esophagus to go through. And when stomach bulges through from there, we can talk about hernia. Hernia can be found in many different places. So it's whenever you have an organ or tissue or part of some sort of a body part that shouldn't be going through an opening and it's going through there. So they can be asymptomatic or symptomatic, but we want to follow that. Gastric ulcers, if the stomach acid damages the lining of the stomach, that can be due, for example, many things, but for example, thanks to a bacteria or high amounts of aspirin. And we want to make sure that we don't irritate that ulcer, so we might want to decrease the stomach acid production until we're healed. Antibiotics if the bacteria was involved. Secretions of the stomach, this is a really good table. We talked about them in quite a detail on our lecture video, so if you haven't watched that and don't feel comfortable with these, I really encourage doing so. What we notice is that the hydrochloric acid, stomach acid secreted from the parietal cell, played important role in making things happen. So we have pepsinogen, which is an inactive form of pepsin, and that gets activated, becomes peptin only in the presence of hydrochloric acid. And pepsin played a role in splitting proteins, breaking them apart. However, we also have secretions in the stomach, gastric lipase, so that plays, remember, lipids, breaks down fats. But that doesn't really happen in the stomach because of the pH that we have within the stomach. So that's going to happen only within small intestines, where the pH is more neutralized thanks to the secretions of the pancreas. So gastric lipase is added at this the stomach, but it's fully activated only in the small intestines. Of course, we have to secrete mucus within the stomach to protect the stomach wall. That's from the goblet cells and mucus glands. We also have intrinsic factor to note plays a especially important role in vitamin B12 absorption, and that was secreted by the parietal cells. So make sure that you feel comfortable with those. We noticed that very little absorption happened in the stomach. We already talked about that. That's thanks to many different things. It's very acidic in the stomach thanks to the hydrochloric acid. So this prevents most enzymes, most digestive processes from working, most absorption from happening because these things don't work ideally in highly acidic environments. So it will be once we get to the small intestines where we have a more neutral pH, where most of the absorption happens. Of course, there's also very little villi within the stomach lining inside. That's an issue. We don't have the great surface area for the absorption, and the thick mucus layer as well plays role, so that prevents some of that absorption from happening within the stomach. Let's look at small intestines. That's the main site of absorption, so that's the business end of what we're going to be looking at. They're about 2 to 3, 4 meters long in length, about 2.5 up to 4 centimeters in diameter. It is really the main site of absorption. Most absorption happens in the digestive system within the small intestines. There's a lot of folds, villi, microvilli here, and that gives us this large surface area. If we spread out the small intestines, remember it's only 2 to 4 meters long. The surface area yet still is gonna be about the size of a tennis court, so a lot more. So thanks to all these folds within the structure of the small intestines. You remember we had our pyloric sphincter. We saw that earlier on here. That's the kind of the start where stomach ends and small intestines start. And then we have 3 parts to know about. We had our duodenum, we had our jejunum, and we had our ileum. Oh, that didn't go exactly as planned. That's better. And the mnemonic that I gave you for those was DJ-IL. So duodenum is the curvature around the stomach. If we see here, this is a little bit of a careless drawing of a stomach, but let's live with that. Right after the stomach, duodenum is this first curvature. Curves around, for example, and I'm trying to change the color of the pen, which seems to be a forever-ending battle for me. That seems better. So it curves around the pancreas that's located here. So that's our duodenum. After that, we had jejunum and ileum. They kind of fill the medial part of the abdominal area. Where jejunum changes to ileum is not clear. That's not really an anatomical change, more of a physiological one. And that all ends, of course, once we get, so this was our jejunum and ileum, once we get to the large intestines here, and the transition point from the small intestines to large intestines here was our ileocecal valve. And that's because the last part of the small intestines was the ileum. So we take the ileo part from that, and cecal part from the fact that the first part of the large intestines is cecum. So names tell often where things are located. We remember there's a lot of secretions in the stomach, but not so much absorption. But a lot of these secretions that play a role in digestion get activated within the small intestines, thanks to the lower pH there. So I've listed some secretions there. Then we have to look quickly through the large intestines. Large intestines don't really play as important role in absorption of nutrients or energy, but it's all about the water and water uptake. If we look at our chyme at this point, that's very watery, very liquidy. And if we were to eject all of that from our body, such as you might, for example, when you have diarrhea, you would lose a lot of fluids. You would have to be drinking a lot to replace that. So it is within the large intestines that most of the water is reuptaken. We reuptake most of the water. And also I think that that's meant to say electrolytes. So let me change that. Electrolytes with the water. You remember when we discussed about the fluid palace. So water plus that's what it's meant to say. So both of those are reuptaken, recovered within the large intestines. We have multiple parts of the large intestines that we need to know. The first part, cecum, we already discussed a little bit about, and I'm again trying to. And so if this was the small intestines here, stomach here, so large intestine started here. The first part was the cecum. From cecum, we have a little bit of extension coming from there. That's known as our appendix. If you do, for example, documents that you're sending to a government office, you might attach an appendix to that. That's something additional, something extra. Appendix can get inflamed, so that can be a little bit of a problem for us. So that kind of brings us to wonder, why do we have something? We don't really need appendix. Why do we have something that can cause us this much trouble? Well, we think that appendix is evolutionary remains. So when we were eating more heavily plant-based diet, the bacteria that help with the digesting the plant-based diet was housed there. So now that our diet has changed, it's slowly shrinking, and evolutionarily will probably eventually disappear. Well, the next part is colon, and colon is made of 4 different parts that we want to know. First, our ascending colon, so climbs up on the right side of the abdominal cavity, it's not as straight, it's more of a bulby structure than what I'm drawing here, but work with me. I'm doing the best I can to draw it. So ascending colon travels on the right side from the cecum up. Then colon crosses as a transverse colon from right to left side at the superior part of the abdominal cavity, comes down as descending colon. And then the last part is known as sigmoid colon, thanks to the shape. That's kind of an S shape that might help you to remember it. And then we have rectum at the very end. So ascending, transverse, descending, and sigmoid. Those were the parts for us to know about the colon. So those would be the parts that I would like you to know. Remember the main function, water recall. Then we have a bunch of accessory organs. I'm not saying that they're any less important. We just spent a little less time on those. Liver plays many, many functions. I once tried to list as many as I could. I think I got to probably 100 and gave up. It is the largest gland in the body, made of about 4 lobes. There's different structures that we've covered. In the class, you might want to have a look at those. Liver sciences. When we talk about liver medicine, we talk about hepatic field, hepatic sciences, hepatic artery, hepatic veins. So that's what we're looking. Really, I think that the big thing about liver to remember for you is that that's where bile is produced. So it's the liver that produces the bile. And that's often something that people get confused, and something that I like to ask in an exam to make sure that you know what you have learned and what you're covering. So bile is produced by the liver. And then once liver has produced the bile travels down the common hepatic duct until it gets to the cystic duct. And cystic duct connects to the gallbladder, which is the structure that we'll have a look next, located below the liver or on the bottom surface there. And then we have bile duct here at the very end, connecting to the hepatopancreatic ampulla here to the small intestines. So hepatopancreatic ampulla here. So that opening is shared by the bile duct and the pancreas as well. So that takes us to the gallbladder, and really remember what I just emphasized, that it was, I didn't mean to highlight that, that it was the liver that produced the bile, but this gets stored and also concentrated within a gallbladder. So gallbladder does not produce bile. That's a common misconception, and something that I think I could very well ask from you. Oh, I didn't mean to do that. Something that I could very well ask from you. Bile is simply stored and concentrated within the gallbladder. You can get gallstones. That's when the cholesterol escapes the bile and forms these solid stones within the gallbladder. Can become a problem, can become something that we need to address. The last accessory structure that we should look in terms of the digestive system is going to be our pancreas. So pancreas is kind of a weird one because it's both exocrine and endocrine gland. And on this diagram here, we see the pancreas. And notice how pancreas opens through that hepatopancreatic ampulla. So we're really interested on this exocrine function of the pancreas, which is, of course, production of the pancreatic juice. Pancreatic juice is the most complete digestive secretion in the body. So plays important role, has a lot of digestive role, also lowers the pH within the small intestines. So the stomach, after the stomach acids, it is the pancreatic juice that neutralizes the pH a little bit and allows most enzymes to start working, ideally function better in that chyme that has passed now to the small intestines. Of course, the endocrine function was the blood sugar regulation, but we're not gonna look at that in this chapter. I do want you to be very, very, very familiar with this listing that I have here. Remember that initially we ate food. Once we've eaten the food and mixed it with saliva, so we've broken down mechanically the food and we've mixed it with saliva, food becomes now a bolus. Bolus travels down from the oral cavity, down esophagus to the stomach, where it gets mixed with our stomach acids and other secretions. It now becomes chyme, much more liquidy than what bolus was. And by the time we get to the large intestines, when we remove the water from the chyme, we are left with feces, the indigestible part of the food that we have consumed, that we then will end up eliminating from the body. So that's the digestive system. And you've seen that I spent a lot of time on this review on that. And that's because it's such an important part of our material. So you can expect that in the exam, there would be a lot of questions about digestive system. We're also going to look through the male and female reproductive systems, but I'm kind of assuming that these are going to be quicker and easier since we have spent recently a lot of time on this. So let's have a look of some of the general concepts that apply both to males and females. in terms of the reproductive system. So the primary sex organ, it's really important that we can tell apart primary sex organ from all other organs within the reproductive systems. And these are known, the proper term that we should be really using is the gonads. And in males, the gonads are testes. So the primary sex organ of the male is testes. And we have a twofold function for the primary sex organs: production of the sex cells and production of the sex hormones. And this is for both males and females. Of course, for the males, testes, the sex cells that are produced are sperm, or spermatozoa might be another term that you see for that. So the sex cells are known as gametes. So male gamete is the sperm. So again, terminology that I do want you to know. The other function was the production of the sex hormones in males, androgens, especially testosterone, to note. A female gonad is going to be our ovary. So ovary produces the egg cell. We talk about ova or oocyte. So that's the sex cell gamete of the female, and sex hormones, the important ones here would be estrogen and progesterone to know. All other parts of the reproductive tracts that we'll look are going to be accessory structures. So only one primary sex organ on both females and males, or they are paired, but only one structure name. So one gonad to know. Let's have a look of the males. We've drawn this diagram in the past, so I'm quickly going to review through it. I have not really filled it in. I just listed words that you should know. So let's review what we should know about this. And I think I've tried to use a certain color on different ones. If I get the colors confused, do look past that. So the male gonad, male primary sex organ, is gonna be the testes. And the function of the testes was the production of the sex cell, gonad, which was the sperm in males, and also the production of the sex hormones, androgens in case of a male, especially the testosterone. After our testes now, and I'm gonna try to change the color. After we're finished with the testes, sperm travels to this structure on top of the testes, and this structure is known as epididymis. This is where sperm goes to be stored, but also to mature. Sperm is not ready yet once it leaves testes, so it spends time here within the epididymis to mature and wait until it's needed. The next structure after that is going to be this long tube, ductus deferens, or ductus deferentia, or vas deferens. All of those are names that can be used about that. This is really a muscular tube that helps us to transport the sperm. So remember, that's the function of this tube. And you'll notice that at the end of that tube, still as a part of it nonetheless, we have this expansion, this larger area, and this is going to be our ampulla. So kind of a dilation of the vas deferens or ductus deferens there. Couple of other tubes to know about. Let's add here ejaculatory duct. Oh, I didn't mean to cross it over. It should work better. So ejaculatory duct is used only by the reproductive system. So there's going to be a bunch of structures that we share with the urinary system and reproductive system, but this first part that we see here is going to be ejaculatory duct, and that's only for the reproductive system. Well, ejaculatory duct receives secretions from a gland, and this is going to be first of our glands that we're going to look in terms of the male reproductive system. And this gland is going to be our seminal vesicle. Ooh. And seminal vesicle secretes a lot of stuff, and these contain, for example, fructose, a lot of energy for the sperm cell to kind of give it another boost, recharge it. If you think at this point sperm has traveled a significant distance, it probably has used a lot of its energy. So this is to help us, to help the sperm at that point, these secretions. The next structure that I do want to talk about that's worth of noticing is going to be our prostate gland. So our ejaculatory duct and first part of the urethra has to go through the prostate gland, and that can be sometimes a problem. In older men, we quite commonly get extra growth of a prostate. I'm just coloring the ejaculatory duct back because it changed colors when I mixed blue on it. So if prostate overgrows, it can shrink these tubes that go through it, constrict them. It might be hard for urination. It might be hard for semen to come through, stuff like that. Then the part after that, let's take the pens out again. We're going to continue our duct system, and remaining part of the duct that we'll see is going to be our urethra. So urethra is shared both by urinary system and reproductive system. So now we are dumping the contents from the urinary bladder, which is located here, to the urethra. So our ejaculatory duct joins the urethra, and material travels down all the way to the opening, penile opening, to the end of the penis, to the external world. So that's our urethra, and we need to know about that. One more gland that I want to mention especially. This gland is gonna be, I didn't mean to change the color here. There has to be an easier way to do colors, but I don't know what it is. The next structure that we're going to have a look of is going to be our bulbourethral gland, sometimes also called Copper's gland. And this gland located here releases secretions in response to sexual stimulation, and again help in formation of the semen. Semen is a mixture of sperm cells and secretions. So something to bear in mind there. And finally, the erectile tissues that we should know. Let's see if we can find a color for them that we wouldn't have used before. The erectile tissues are going to be our corpus cavernosum and corpus spongiosum. I'm trying to highlight them a little more so they show up a little bit better. So they are these erectile tissues here that help us to harden the penis so that it's easier to insert that into the female body. And I'm not expecting you to know too much more about the corpus cavernosum and spongiosum, but that would be kind of nice if you know that they're there. So that's about it about the male reproductive system. I had just a couple of other mentions about that. The structure, scrotum, was the structure, the muscular sac where the testes are located. That sac is outside the body so that we can lower the temperature of the testes a little bit lower, about 3 °C lower temperature than body temperature is ideal for the sperm production and maturation. So that's why the scrotum exists. If it gets cold, scrotum pulls up thanks to the dartos and cremaster muscles, and we make sure that it doesn't get too cold. Sperm cell, we're going to look at different parts of the sperm cell. That's going to be something worth of knowing. So we've divided sperm cell into 3 parts. So the first part was the head part. Within the head part of the sperm cell, we house the genetic material, so the DNA that we want to pass to the female egg from the male. And in addition to that, the head also contains some digestive enzymes that help us to make our ways, helps the sperm cell to make its way to the female egg through the outermost layers of the egg. After that, we've got a mid piece, which is this kind of a part in the middle after the head, before the tail of the sperm. And this part is rich in mitochondria. When you ask yourself, why would we have a lot of mitochondria here? Think of what sperm is going to be doing. Its tail part is going to be doing the locomotion, so the moving part. So we want to have a lot of energy for that, and mitochondria serve to produce that. So that's why it makes sense to have a lot of mitochondria here, so that the tail can have resources for the locomotion. So those were the 3 parts of the sperm cell that we've kind of covered quickly. I've drawn them there. In terms of the hormones, I've talked a little bit with you about the hypothalamic pituitary gonadal axis. So all of hypothalamus, posterior pituitary, and testes do their own secretions effect as a bundle of 3. Let's review that if you don't know. In terms of the testosterone levels, you'll notice that there's high amount of testosterone during the pregnancy. This is involved with the sex selection and some initial changes due to whatever sex we end up with. After birth, just a tiny jump in the amount of plasma testosterone in the blood, and then it remains fairly low until you reach puberty, where we have a significant spike in the amount of testosterone, and that remains high up to your old age, where there's a decrease once you hit past 65 or so. Same in terms, I think that's a pretty good diagram. Sorry, I take that back. About the testosterone, I'm not going to expand further. I don't want to add more than you have to know. Let's have a look at the female reproductive system again. Here I have a diagram that should be familiar. We've already discussed that a little bit in our previous classes, so let's see what we can find. So the main sex organ, so the gonad of the female reproductive system, is gonna be our ovary. Ovary produces the female sex cell, so oocyte or ova, if you wish, and of course the sex hormones and androgen and progesterone. After the ovary, when egg cell leaves the ovary, egg cell actually leaves and sneaks outside the female reproductive tract to this pelvic cavity region, and it just floats there free. This egg cell is not passing directly from ovary to the uterine tube, because there's no direct connection between the ovary and uterine tube. Instead, it spends a little bit time on this cavity outside the reproductive system and then is caught and taken back to the female reproductive system by a structure that we're gonna call fimbriae. So fimbriae are, if you wish to describe them somehow, a little bit like these finger-like structures that are the first part of the uterine tube here, and their function is to catch the egg cell from this pelvic cavity and direct it to the uterine tube. So that's the function of the fimbriae. No direct connection from the fimbriae or between the fimbriae and ovary, but it does catch the egg cell. You'll notice immediately after fimbriae, there's this part that kind of looks like a cone-shaped part of the uterine tube. This is known as infundibulum. Infundibulum simply helps us to direct the egg cell from the fimbriae. I don't know what I did there. Helps us to direct the egg cell from fimbriae to the uterine tube. So it was this kind of a cone-shaped part here distally in the uterine tube that we should know about. After that, we're going to continue our journey and we're going to look at the rest of the uterine tube, sometimes also called fallopian tube. So both of those terms you might see they are pretty much interchangeable. Depends a little bit of the concept, depends on the textbook and so on. So the uterine tube has two parts that I'm going to want to highlight to you, especially. Let's get a pen for doing that. So first of all, this very first part, longest part of the uterine tube here, kind of you'll notice it's a little bit more wiggly. There's a little bit more curvature there, a thinner wall. This is known as ampulla. Within ampulla is where the fertilization takes place. So egg cell has traveled there and sperm cell has traveled there. And this is where the fertilization takes place. After that, the remaining about 2 centimeters, much thicker wall of the uterine tube is known as isthmus. And isthmus connects to the uterus. See. And for the uterus, I'm going to want to highlight it with 2 different parts. And let's start with the roof of the uterus, which is going to be fundus. We've heard the word fundus before, the roof part, superior part of some organ, or of an organ, of a structure. So fundus is the roof of the uterus here. The majority of the uterus, about 2/3 of it, is made of body. So let's add body here. Body of the uterus. And the remaining about 1/3 is gonna be our cervix. And this is where we would take, for example, the cervical sample if we want to check for that. And notice how cervix protrudes a little bit to the vagina. Partially. And then we have a couple of external parts that we might want to notice are vulva, peritoneum. I don't really have them on this diagram, but check those out if they're unfamiliar to you. Where do we want the fertilized egg cell to attach to? That would be a good question. Well, we want the egg cell to attach to either to the body of the uterus or to the fundus. If it attaches to the cervix, it's not necessarily an end of the world, but that does carry a risk factor. So we would monitor that pregnancy more carefully. We would also want to be aware of that when the time for delivery comes, in case the placenta forms and blocks this opening of the cervix. That could be a problem if placenta deattaches when the baby has not been born yet, and we have an issue with that. A couple of ligaments, I'm gonna leave them for you to review independently, but remember we had our ovarian ligament, our suspensory ligament, and our broad ligament. So we can add all of them to this diagram, but you can do that independently. There's already a lot going on there. In terms of the female reproductive system hormonal control, there's a lot to look at as well. We just went through this sheet in the past, so I'm going to go through it real quick, but this is something that I would kind of assume that you're already familiar with. So everything starts with the hypothalamus that then controls the anterior pituitary gland, which then secretes gonadotropins. First of the gonadotropins that we're going to look at is going to be follicle stimulating hormone, so FSH. And follicle stimulating hormone helps with the development of the follicle, which is this structure that contains the future egg cell and the cells around it within the uterus, within the ovary. And the follicle develops until the egg cell is ready, and at that time we'll see luteinizing hormone spike, again from the anterior pituitary, which triggers the ovulation to happen. So now the egg cell is shed. Well, what happens is that the egg cell starts its journey through the uterine tube, catched by fimbriate, and then travels through the infundibulum to the uterine or fallopian tube, and remaining part of the follicle is left behind. This becomes corpus luteum, the remaining part of the follicle, and corpus luteum starts to secrete estrogen. Remember, this is when the egg cell has been shed. We are preparing in case the egg cell gets fertilized and the individual gets pregnant. So what corpus luteum and the estrogens secreted by it does, it promotes the thickening of the uterine wall line. So the uterine wall starts to grow nice and thick. So it's good for the fertilized egg cell to attach to, and it will be able to support the fertilized egg cell. Another thing that's secreted by the corpus luteum is going to be progesterone, and what progesterone does, it makes the uterine wall lining that's thickening also more vascular and more glandular. So all of these purposes help us to make the uterine wall ready for the implantation of the fertilized egg cell that we hope that it's going to happen. So you'll see it thickening here thanks to the estrogen, and then becoming more vascular and glandular thanks to the progesterone. Well, what happens if there's no fertilization? Let's start with that scenario. Well, if there's no fertilization, the corpus luteum will degenerate, and when corpus luteum degenerates, it stops producing estrogen and it stops producing progesterone, and as a result, the uterine wall lining will not be maintained, and instead it will be shed as part of the menstrual bleeding. So that's how we get rid of it. So that's what happens if there is no fertilization. Luteum disappears and estrogen and progesterone secretion increases, causing the menstrual bleeding, getting rid of the thickened layer of the uterine wall. If there is a pregnancy, the corpus luteum gets salvaged and the secretion of estrogen and progesterone continues, and there is not going to be menstrual bleeding. And we maintain the uterine wall lining for the developing life to develop there. So that's a very good quick summary of what happens. In more detail, you can review it from our full lecture notes. Couple of last terms that I do want to mention. I want to mention about the menstrual cycle. The proper term that we're going to use for talking about menstrual bleeding is going to be having menses. And the first menstrual cycle is going to be called menarche. We're noticing that the first menstrual cycle is happening a little bit earlier than in the past. There's a little bit of speculation what causes that. Menopause is when menstrual periods have ceased for 1 full year. That's when we consider that menopause has happened, or a woman has gone through menopause. And there were a little bit of more detail in the lecture video about that. Another thing to mention about the mammary glands, of course, both males and females have those, but they're only functional within the female. The function of the mammary glands, I would assume that you all are familiar that the function of the mammary glands is the production of the milk for the baby after the pregnancy has ended. Humans are one of the very few species that keep drinking milk, or cow milk in this case, after infancy, and other species milk. So humans are a little different in terms of that. Milk and rather than milk replacement has many benefits. You can kind of look into that yourself, and same with the screening for breast cancer. So that's all that I have as a review video, or a review of the study guide. I think that there's a lot to go on there. And I'm going to jump back into our PowerPoint with hopes of it working. This looks better. So just to kind of recap what we have just done and what's important to remember for this week, we just reviewed the study guide for the exam 4. So I hope that you found that helpful and useful. The study guide with the blanks is available on your Canvas course. I reminded you about the fact that there's no new assignments, but please work through any missing, incomplete assignments. This week would be the time to finish any of those. So I really want you to try to get those in. Once the course ends, I'm not going to be able to accept those anymore because the grades will have been submitted. So finish any missing or incomplete assignments during this week. This would be a great chance, since you don't have anything extra in terms of the work other than the exam and practical to complete. So there should be no reason why there's no time. Remember, our exam is going to be open book, and you can take it wherever you wish. No proctoring, so you don't need to have the video camera on there. You can use your notes. It is timed. 60 minutes if you don't have an accommodation plan. If you have an accommodation plan, then it's 120 minutes, so 2 hours. But really try to answer to every question, try at least to guess. And there is an extra credit question as well. So that's a chance to earn extra points as well on that. And I will try to go through those as soon as I start getting them in. Like I said, try to make sure that you get the exam and lab practical done by the end of this week, end of the Sunday day or Sunday night. If you miss, I won't go crazy. It is possible to finish them also at the early part of the next week, but really that's pushing it tight. So try to get them in as well as you can during this week. We've reviewed the study guide. There's study guides also in your lab for your lab practical 4. There's the practice lab practical 4. I encourage you to use that, review that a couple of times so you see all of the structures that might be asked. I said that I will upload handouts if they're not already there for each chapter. And like I said, make sure if you want to retake or have something opened, missing, or an assignment that you don't feel that you did that well on, I am happy to facilitate that, but time is going to be of an essence. We're kind of running out of the time for all of that. So send me a message if that applies to you. But this really needs to happen soon to get these in, because like I said, I have to get the grades finalized at the end of the finals week. So send me a message if they're on Connect, if there's anything you want reopened, or you want it has closed, or you did not score as well and you want to reattempt it. Goes for both for assignments and exams, practicals. So that's something to bear in mind. My goal is to get you those points. There's going to be extra credit, like I said, also on the finals week project. And I will share the details of the finals week project next week. It's not going to be memorization. It's just a project that you do. And the tutoring is available through the college. Also, I'm available for you if needed. I don't think we have any questions this time. But that completes everything I had to share with you on this week's review session. As always, if you have questions, feel free to contact me. The easiest way is through the Canvas messages. And if you don't hear back, just message again. I am trying to make myself available as much as possible for you. So that's all for this video. I'm going to wrap up now. We're going to have another session on Wednesday morning at 10 o'clock and Wednesday evening at 8 o'clock p.m. So, and you have this recording also available. So that's all for this review session. Thank you for joining it live or the recording.